Firstly, I'm going to introduce the structure of the DNAs. There are two strands in one single DNA. In the three main elements of a DNA is deoxyribose sugar, phosphodiester bonds, and the nitrogen bases. And there, in the sugar, it's formed by 1, 2, 3, 4, 5 carbons and one oxygen, and the phosphodiester bond just connect two sugars. There are four kinds of nitrogen base in DNA. It's adenine, thymine, guanine, and cytosine. They are corresponding. For example, in one strand, if it is thymine, on the other strands, the corresponding one should be adenine, and the cytosine and guanine is corresponding also. And there are three hydrogen bonds between the cytosine base and the guanine base on the two strands, and there are only two hydrogen bonds between the adenine base and thymine bonds base on the two strands. Okay. Secondly, I'm going to show the process of DNA replication. Firstly, the helicase is going to start to cut off the hydrogen bond between the nitrogen bases. It will start at this point because there are two G and two C's continuously, and I put it here as it going towards the two direction. It will cut everything off, and also this part of it. And after cut it off, the RNA polymerase and DNA polymerase is going to show up, and RNA polymerase is going to produce something that can attract the polymerase in order to lead his way, and after this go all the way down, and the other side, same thing in the other side, they're going to read the information of nitrogen base and to copy it to make the corresponding one. For example, this is G, and it's going to make C, and T, C, C, A, C, C, C. In this side also G, G, T, T, G, G, A, G, C, C, G, G, C, and C. And then they're going to start from where this began from. But the only difference is they're going to the opposite direction, which is this side, and also G, G, C, C, G, and G. And after this, lingus showed up. They start here, in here, and then what they do is they produce the bonds who glue and get the things together again between all the new DNA strands and the old one. So in the end, actually, the one single DNA became two DNAs, and each DNA contains one old strand and one new produced one. Next, I'm going to talk about the process of protein synthesis. You can see that this is the zoomed in vision of a cell, and this part is the nucleus of the cell. There are a lot of DNAs in the nucleus, and in order to produce proteins, the information on DNA has to be read. But the problem is the holes on the surface of the nucleus is too small for DNA to get out. So the transcription is needed. The transcription is very similar to the replication of DNA, because just like replication, the hydrogen bonds of the DNA will be break down, and then the transcription process started, and to produce a messenger RNA, which have the a corresponding nitrogen base to the read one strands of the original DNA. In other words, the nitrogen basis on the messenger RNA is the same as the other strands of the DNA. And now, since the messenger RNA only have one single strand, it is small enough to travel through the hole out of the nucleus to the other part of the cell, and the protein synthesis happens exactly out there. And the nitrogen basis on the messenger RNA is divided in the group of 3, just like this, and each of them is called codon. And as the RNA is read in this part of cell, it's called ribosome. In ribosome, the tRNA came in, and tRNA contains each of the tRNA also contains 3 of the bases, corresponding bases of the codons on the messenger RNA, which is called tRNA. And also, the 3 of them is called anticodon. Okay, the tRNA is all the stuff here. And then as it go in, each of the amino acids on the tRNA will stick on the others, which is produced before that, and they stick on that to add new amino acid to the row. After that, it leaves the ribosome with no amino acid. And then, in the end, the whole long row of the amino acids is called polypeptides, and in the end, the long row of it forms the proteins. And last, I'm talking about the mutations. Firstly, as a DNA is like this, it's a normal DNA, and it's transcribed into messenger RNA, and according to this messenger RNA, the amino acids and therefore then the proteins are produced. But what if mutation happens on the DNA? For example, the A is replaced by T, and then the corresponding RNA, the codon, is changed, and therefore at last the amino acids this codon produce is different. But this mutation is called point mutation because it only changed one single base on the DNA, and it will cause only one single difference in the role of amino acids. But what if one of the base on DNA is replaced, is just took out, or we add one more on the strands? Or also, it's possible that the whole codon is taken out, or they add a new codon in this. So in this, the only single base are replaced or were taken or added in, the whole grouping is different. So the information is entirely different. So every single amino acid this produce will be different, and this is called the frame shift, the mutation, in which it can cause a lot of problems or maybe benefits. And also the physical mutation can also happen, and normally the DNA is rolled like this, and it cannot be read. But in the very early times of people and human or any other animals, the DNA will be unrolled and then read to form all the proteins and organisms. And then it's rolled again, and that will never be unrolled and read again. But what if it's some chemicals or vitamin A or something like that cause the whole DNA to unroll again, then it be read again, and the new proteins and new organisms will start forming, and that is physical mutation. And the mutation sometimes is benefits and the other time is bad. When it's bad, then the organism, or possibly human, will have no time to reproduce. and if he will have, or he or it will have no offsprings, and this kind of mutation cannot be passed off. And if the mutation benefits the organism, and it will have more advantage in his environment, and then he will have more time kind of to reproduce, and he will have more offsprings, and then this kind of mutation will pass off, and then finally became the normal DNA. And that's how natural selection play a part in this. And without mutation, there will be no new kind of features or whatever it happens in all the generations of an organism or species. And the new feature, without new features, there's no evolution, because every generation will be all the same. In addition, the external causes like radiation and chemical elements, and internal causes like the errors and mistakes made in DNA replication will all cause the mutations in DNA.